Are we ready? Okay. So welcome everyone to Grand Rounds, and I welcome everyone at Harborview who are watching us from their conference room, and of course to our colleagues at Seattle Children's who join us every week from their conference room as well. So welcome to all. And I have one bit of business first. So on November 13th at the faculty meeting here, we are going to have a presentation by Marie Carter-Dubois, who's Director of Business and Operations for the School of Medicine, who's going to be talking about details of a program they're unrolling generated by School of Medicine to help us with pre-award administration, so sending in the grants. So this is different from all the other people with whom you've worked when you're sending in grants. This is a new team. Their only focus is pre-award, and they seem very committed to working with us, and I think they're going to do whatever they can to get us our grants, aside from shifting pay lines. So we have our final lecture, and Dr. Kreutzfeld's produced 5 New Things series, and so you know our next speaker as our chair of the department, but also he is, of course, world-renowned as an influential researcher in neuroscience and neurology, and as we have heard many times, more than 25 years ago, began a journal called GLIA, which presaged the research advances in non-neuronal cells. And so he was interested and had an intuition about these glial cells before they became the sexy little beasts that they are. So in addition to being the editor-in-chief of GLIA, he is also on the editorial board of numerous neuroscience and neurology journals. He has been a long-standing NIH-funded researcher. So I think it's clear that Dr. Ransom has his finger on the pulse of neuroscience and neurology and what's important among what is new. So, Dr. Ransom, thank you.
Good afternoon, everybody. Sumi is on the brink of being too eloquent of an introducer, so she leaves the speaker in an awkward position of having to follow her eloquent comments. So be careful about that.
I would come just for the intro.
I'm sorry?
I would come just for the intro.
So I'm going to pay very close attention to how I project my voice, because I want our friends at Harborview, hi there, folks, and Children's, to be able to hear everything. And apparently that's a little bit position dependent. So 5 new things in clinically relevant neuroscience. That posed some challenges for me. I want to start by addressing the issue of whether I might have a conflict of sorts. And I mentioned that one of the things I'm going to talk about is a product of a company called Aeromics, and I'm an unpaid consultant. Who knows, Tom? Maybe someday to be a paid consultant, but I can't say that at the moment. The only other possible conflict of interest is that I might have had more fun putting this talk together than you're going to have listening to it. If that turns out to be true, I don't want to. Don't tell me about it. Better that way. So as I mentioned, my challenge was simply that I can't really think of anything in neuroscience that isn't relevant to neurology, and that meant that I had quite a playing field to think about in terms of what exactly to pick as topics for our conversation today. In doing a little looking around, I certainly encountered some wonderful things that I would encourage all of you to take advantage of. And first and foremost among them is an issue of the journal Neuron, which is a wonderful, very prestigious, very high-impact journal accepting only the very best in neuroscience. And about almost exactly a year ago, in fact, I guess a year ago, October 30th, 2013, they published an issue which basically celebrated 25 years of this journal. It is jam-packed with wonderful survey articles about what I would have to admit is really the full spectrum of neuroscience. And I didn't take anything from that journal. And of course, glia, yeah. Okay. Now, in trying to get some inspiration for this, I did look around, and wasn't this interesting? Emerging Cognitive Neuroscience and Related Technologies. This is from the National Research Council Committee on Military and Intelligence Methodology. So that sounded kind of intriguing, and as I read on, they, in this joint commission, and they published all this stuff, they were exploring the implications for United States national security of certain things in neuroscience, things that should be monitored by the Intelligence Committee. So pay attention. Some of the things that they're interested in thinking about are neurophysiological advances in detecting and measuring psychological states and intentions of individuals. So this, in fact, we did a little skit at a resident graduation not so long ago, and Bill Spain wore a contraption because he was worried that the government was attempting to read his thoughts. They are trying to read your thoughts, Bill. Now, I don't know if they're trying to read your thoughts exactly, but that's on their game plan. They've thought about the development of drugs or technologies that can alter human physical or cognitive abilities. They'd like to be able to examine Bill's brain in real time. And of course, they're interested in artificial intelligence. And remember, they're going to be monitoring this. So there wasn't too much there for me. So I settled on these 5 topics. These are not in any way, shape, or form equivalent, as you'll see, in terms of how I'm presenting them. And some of them are going to fly by pretty quickly, but I'll do my best to make the few slides on some of the topics as relevant and clear to you as I possibly can. If you'll do your best to stop me if I'm saying things that you don't understand. So let's get started. This is somebody that you haven't met, but you have probably seen his picture, Peter Agre. He won a Nobel Prize in 2003 in chemistry. And he discovered, and he's been very almost gleefully exposed about how he came about to make this discovery, which he claims, I'm sure correctly, that it was purely by serendipity. He was interested in Rh factors, and he was studying red blood cells, and he kept finding an annoying protein in some of his assays that had a molecular weight of 28 kilodalton, so they called it 28K, and it had other interesting names. A completely undiscovered molecule, and it turned out that he was eventually able to demonstrate that it was a water pore, the first of its kind. And this water pore is everywhere, not in every cell. Some cells are loaded like a red blood cell. The water pore permits water to go back and forth, and to do so without dragging ions with it. We have lots of other pores, channels, sodium channels, potassium channels. They take water back and forth, but not in great quantities and always with other things. So this aquaporin was a stunningly interesting molecule. And he published that first report in 1992, and a mere 11 years later, he got a Nobel Prize in the mail. He did a lot of wonderful work, X-ray crystallography, and was able to demonstrate what a unique protein it is, et cetera, et cetera. But oh my gosh, maybe a problem. A Romanian scientist stepped forward and said, hey, wait a minute. I published my paper on aquaporin about 2 years before you published your first paper, and I'm really upset about that. You can see he looks upset about this. And he has apparently appealed widely to the Nobel Committee and to Peter Agre directly. I don't think they have a real intimate and warm correspondence, that maybe he should share his Nobel Prize because of this paper of his. Frankly, I'm making a little light of this. The Nobel Committee has made egregious mistakes in the past, and this may be an example of another colossal mistake. We simply don't know yet. Aquaporins, I think, are going to be incredibly relevant to those in the audience who do neuroscience and those of you in the audience, a lot of you who practice neurology. And that's really the purpose of the next 5 minutes. So we don't have great treatments for stroke. I think that's an understatement, but it is certainly something we've learned to live with, and it probably bothers us less than it should. One of the things that we completely ignore about stroke is that almost inevitably it causes a huge amount of swelling. Most of this swelling has been referred to as cytotoxic, meaning that it's cells that swell. People have estimated that upwards of 50% of the morbidity and mortality of stroke may be related to this swelling. And again, it's on the list of things that we simply can't treat. Although my comments are going to be particularly directed to stroke, as you listen to them, think about this possible agent in the context of your clinical interest, because you might find it surprisingly relevant. So we were talking about edema, the two forms, vasogenic, the blood-brain barrier breaks down, blood is able to enter the brain or an ultrafiltrate of blood, and then with cytotoxic edema, and these terms are quite old and are probably not quite appropriate today, but they're still in common usage, and therefore I'm using them here today so that this sounds a little bit familiar to you. Cytotoxic edema, the swelling of cells, probably more than anything else involves astrocytes. One of our speakers quite recently, Brian McVicker, is working on a story which seems to indicate that against all of the published literature, neurons in certain areas of the brain may have an incredible capacity to swell and may do so to the extent that it adds to the morbidity and mortality of certain conditions. But for today, and since I've got the podium, and since I like glial cells, I would like you to think that astrocytes are the primary villains when it comes to cytotoxic edema and stroke. So one possibility might be, because these aquaporins are very, very densely expressed on astrocytes, which swell, one possibility might be that if we could block the aquaporin primarily expressed in brain, Aquaporin IV, that maybe this would do some good, reducing morbidity and mortality of stroke. It makes a lot of sense. There's some theoretical stuff to indicate that with very toxic compounds that in fact can sit in the water pore, there are certain heavy metals like mercury that can do this, but I don't think anyone here is going to be immediately tempted to use mercury as a therapeutic agent. So a nice theoretical idea. And in fact, it's more than theoretical. In today's day and age, we can make animals that don't express Aquaporin IV. So when you do that, and you expose the animal to an artificial situation of water toxicity, you load them up with a ton of water, and their brain swells. And we do this in animals that have normal amounts of aquaporin, and you can see that most of them rapidly die. When you use your knockout animal that no longer expresses Aquaporin IV, primarily in brain, these animals do terrific. It's a spectacular rescue. The same is true in stroke. When you take and compare ischemic stroke in animals between a wild-type animal expressing normal amounts of Aquaporin IV versus this knockout animal, I don't think you need a magnifying glass to see that there's a lot more swelling shown here by this pale tissue in the wild-type animal than in the Aquaporin IV deficient animal. And when you look at the outcomes, death included, there's a big benefit in favor of the animal not expressing aquaporin IV.
Do the knockouts have any problems at all?
They have almost no problems, Phil, but that's a little bit more of a complicated answer than I'm prepared to give you. They are not entirely normal, but they breed, and so long as they don't encounter a scientist bent on making them water toxic, they tend to do pretty good. So great. This is easy. This is easy. This looks very impressive. We need a drug, and we should just be able to go and look through a big catalog, a medicinal catalog, and find a drug that blocks Aquaporin IV. And Annie and I, who's one of the residents I'm working with this week, would have that drug in our pocket when we saw our next stroke patient. But there aren't any. So that's a problem. Peter Algray and some of his colleagues said there will never be an Aquaporin IV channel blocker because the shape of this protein is such that nothing that a human being would want to take in is ever going to be designed to fit in that pore in a highly selective fashion. And that was a long time ago, and he's been right pretty much until now. So a company called Aromics, headed up by a group that used to be at Yale and is now primarily not at the Cleveland Clinic, but at Case Western Reserve University in Cleveland, has been working on the idea that maybe you could find an Aquaporin IV selective blocker. And I give these guys an enormous amount of credit. From the start to the present, probably a full decade has elapsed or close to it. And they have something that is stunningly exciting. I can show you these slides, some of which I have been given by Aromics because they have worldwide patents that protect this product. So Zusheng, put away your iPhone to take all these pictures so that you can start your own company. They made a very clever assay, and their assay was simple. They made cells swell, and then they could look at hundreds, literally thousands of little test tubes with cells in them that were exposed instantly with a robotic kind of laboratory tool to hyposmotic solutions. The cells swell when they burst. And you put at the same time a drug that you're interested in testing into that well to see if anything changes. And if you do this over and over again, occasionally you get a positive hit. A cell that's seen that exact same hyposmotic challenge, and it doesn't swell and it doesn't die. Now, if you're going to go about this business, you have to be patient. They screened 161,000 unique small molecules, almost all of them pre-screened for at least the remote possibility of being human compatible, druggable in pharmacologies. I don't think that really goes very good, but you know what I'm talking about. So they found some compounds. They found a bunch that belonged to a class of medicinals called phenylbenzamides. And then they whittled this down based on all kinds of things, including potency, toxicity, et cetera, et cetera. And they had about a dozen compounds out of this gigantic group. And then they found one that was clearly the lead compound. It wasn't very soluble. So that took them another couple of years to figure out how to make a prodrug that was very soluble, continued to be non-toxic, and when delivered would eventually go back to the agent of choice, which is AR270 or AR271. I sometimes get a little confused. And it turns out, ladies and gentlemen, that this is an exquisitely sensitive blocker for Aquaporin IV. This is an assay that looks at toxicity at different concentrations of a drug. So here's a low concentration. Here's an extremely high concentration. And when we test this pro compound, their lead compound in humans, you can see that in comparison to the concentrations at what it would be used, it is orders of magnitude away from what would be even remotely toxic. So that's looking good. Now it comes down to actually testing the compound. They use those same models that I was talking about, making an animal develop severe cytotoxic brain edema, and they tried this assay, which is percent survival. Without the drug, most of the animals promptly die, and with this drug, there's an enormous amount of salvation. Most of the animals do extremely well. That looks terrific. They went to a stroke model. This is middle cerebral artery occlusion. And in that stroke model, after the drug, and they've done probably in the neighborhood of hundreds of animals under extremely stringent conditions. Without the drug, there's a huge amount of edema. With the drug, there is a great deal of prevention of this cytotoxic edema, all reaching very clear statistical significance. When you look at neurologic score, and you can do, it's not a great examination. Mice tend to have very little to say at the bedside, so you don't have to really investigate their speech too much. But you can do a pretty quantitative neurologic examination, and when you do that, this drug is a winner. So that's the momentary end of the story. The company's name is Aromix. I am not a principal in the company. I've been advising them for maybe 5 or 6 years. I have never been as excited about a compound that will soon enter clinical trials at a very early stage as I am about this compound, and primarily because I think the potential impact is monstrously large. I think stroke, severe meningitis, encephalitis, trauma, lots of things. Yes?
Thought maybe in this day and age, somebody would have tried to make a monoclonal antibody against the aquifer, or something that would close off the port. Anybody tried that?
They've done things like that, and it is possible to do that to some extent. The problem is access to the brain in sufficient concentrations where it would be effective there. If your intent were to get to Aquaporin IV on a different organ, there is some Aquaporin in the Aquaporin IV and lots of other Aquaporins in the kidney, you'd have pretty good access. But proteins getting into the brain in a wide enough distribution to make you confident that things are going to work hasn't been a particularly valid or useful strategy. I didn't mention, and obviously I've left out lots of little details in the story that I've just told you, this compound can be given after stroke. It has very good access to brain, and the results continue to look absolutely terrific.
Bruce?
Yes?
The relation with the antibody that Aquaporin IV and neuromyelitis optica, is this, I don't know exactly, I don't know the physiology of that, but is there any relation to what's blocking the water?
Yeah, well, so that's a good question. John asked a question about Aquaporin IV antibodies and neuromyelitis optica, Devic syndrome. The truth is that has been flogged as the pathogenetic feature of neuromyelitis optica. I don't find a lot of the data very convincing. There's no question it's highly associated with neuromyelitis optica, but I think it's a marker. It's never been explained to me, even by the people publishing in high visibility places, exactly how it would induce the disease in the places it does. Aquaporin IV is spread throughout the brain almost with precision density, which is uniform throughout. There's not an extra special helping in the optic nerve or spinal cord.
I have a question.
Yes?
The implication is that the aquaporin space is a two-way channel. If you're shutting it off in cerebral edema, which you would not be approaching until it was a clinical issue, what are you doing to the question of having the trapped water inside the neurons be able to escape?
Yeah, so I was just asked a question about whether or not this could be a double-edged sword. In medicine, we're surrounded by double-edged swords almost every day and in every direction. I would say that that's a valid point. I think the belief here is early treatment is going to be key. Once that brain gets to be the size of one of those little mouse patients I showed you, then Aquaporin IV inhibition is probably not going to save the day. So I think that there's some justification. It's pretty clear that edema develops gradually over time. We've got stroke people like John in the audience who would be able to tell us chapter and verse about, on average, it's maybe 72 hours or something like that. I think there would be a terrific opportunity. This channel is primarily on astrocytes and primarily around blood vessels in astrocytes. So it's got a very unique locale. And water could continue to move in and out from cell bodies and other portions of the cell. Okay, let's go on and talk about brainwashing. I'm trying to be humorous here. Come on, you guys, work with me. It's 4:30 or a little bit before 5. I realize that, hey, and some of you in the audience have always wondered, what the hell is the Virchow-Robin space about? I know Patricia's been in my office many times, almost in tears. I can't take it anymore. If I hear that term one more time with no explanation, that's it. I got you covered. This is wonderful work. We've had at least a couple of lectures that have touched on different aspects of the work. I'm going to roll it up very quickly. Micah Nedergaard is coming herself, and she's really this spectacular, vital force in neurobiology who has contributed so many completely unique ideas that I think are going to have clinical significance and certainly have caused an enormous amount of excitement in basic neurobiology. One of them that I'm going to talk about today, actually two of them I'm going to touch on, but the one that deserves a lot of recognition for her input and inspiration is this story about CSF. So for a long time, Micah, who's also trained as a physician, had as many as 24 postdocs in her laboratory. One day she had a student come in who was reading something for a class talking about CSF, and it was immediately clear to Micah that nothing had changed in how CSF physiology was taught for something like 50 years. Now I'm exaggerating a tiny bit, but not much. And as she looked at it, there was something about it that didn't make sense. So she asked the student to engage in a set of experiments using her fantastically powerful imaging techniques. And she said, why don't we look where fluid actually goes when you put it in the extracellular environment of brain? And so what she proceeded to do with her students is put dyes of different molecular weight. This TRD3, I don't remember, it's a middle size, it may be 1,000 Daltons or something like that. Then there's a big one here, FITC. This is something like 10,000 Daltons, much bigger. Put in dyes of different strengths, put them in the cisterna magna space, and then using imaging techniques, and they make a mouse, use a sterile surgical technique. They put a little cranial window in. They attach these incredibly powerful microscopes and imaging things so that they can follow the course of the dye. Everybody with me? So these are living animals. They're running around in a cage. They're quite happy. They don't pay any bills. They've got great care, and things are pretty good. And then she's able to follow where this stuff goes over time. They add another dye so that they can stain vessels and distinguish between veins, shown here in V, and the branches of veins I've circled in blue to make the picture a little bit more intuitive, and arteries. And yes, I did circle the arteries in red. So that's the picture. This green stuff coming in, just for you, Patricia, the Virchow-Robin space. So the Virchow-Robin space is the way that stuff goes from the subarachnoid space into the depth of the brain, and it goes way into the brain, and it gets there so fast it makes your head spin. The big ones stay in this periarterial space. They don't go anywhere else. But the smaller dyes disappear rapidly. And that's what this story is all about. When you look at depth, you see the same thing. Okay, now you've got the idea about what she did. Now I'm going to tell you what she found. She put those dyes in, and I'm going to use cartoons to make things a little bit quicker. And the dye goes down the Virchow-Robin space into the substance of the brain, all the way into the brain, the forebrain. You can see it tracking along the arterioles all the way down. If she waits a little bit, she begins to see the stuff coming out of the brain around the veins. So it is passing from the Virchow-Robin space around arteries to a somewhat analogous space around veins, and it's leaving the brain. Pretty cool. And she named this, and you'll understand why in just a moment, the Glymphatic System, which is a combination, a hyphenation. There's a certain English term for when you add two words that don't really normally belong together to come up with something that's a little like lymphatic drainage, but it's mediated in a very important way by an aspect of glial physiology, which I have not told you. So I'm withholding the secret ingredient. Here's how it works. The stuff comes in on the arterial side. It is believed that this stuff actually is pushed through the space by arterial pulsation. If you change blood pressure, increasing it or decreasing it, you change the amount of the flow and its speed. This is not sluggish, ladies and gentlemen. The stuff whips through your extracellular space of brain with a lot of activity. It moves fast. It removes things. She's done a lot of experiments to show that it picks up proteins that you probably didn't want to have there. It depends on Aquaporin IV. And it depends on Aquaporin IV probably because the Aquaporin IV is loaded on the end feet of astrocytes, which completely encircles these arterial branches going into the brain, especially more deeply in the brain. And here's the proof in the pudding. When you look at what the brain looks like after about an hour of this infusion process, the whole macroscopic brain, it gets a lovely green color, and in fact, in real life, much brighter than this. But if you use that knockout animal that we were just talking about, very little of this gets into the brain. So the first passage of this stuff, I don't believe that it's the dye going through the astrocyte. I believe that water is going through the astrocyte and providing this convective current of water being driven by arterial pulsation to drive CSF in a cleansing action through the brain. Come on. And this cleansing action is shown here. Lots of things. You know what? We're terrible housekeepers. Your brain is filled with junk. Proteins break off. Some of them had a purpose, an hour ago, but now they're just junk, and you toss them in the extracellular space. There's no landfill in the brain that we know of. So you just chuck it out the window, out of the membrane, into the extracellular space. And did you think someone was actually going to come and take your trash out? Well, that's been a kind of a mysterious process. It turns out that if you put some in and tag it so you can follow it, this stuff disappears quite promptly, except in those animals that don't have Aquaporin IV. Then it just hangs around. She used, for drama, beta amyloid, because beta amyloid has a bad reputation of hanging in your brain over many decades and giving you Alzheimer's disease. But this would probably be true for a whole bunch of other things that you would prefer not to have around for a long period of time. Okay, this really should be two stories because I'm shifting gears right now. I've told you something that completely refocuses the discussion about cerebral spinal fluid. The idea that it's literally washing individual cells in your brain, doing it all the time. Here is an incredible kicker. This process happens with your sleep-wake cycle. When you're up and active, like we are right now, your extracellular space is very significantly tighter than when you go to sleep at night. And I'll show you, I'll dramatize that in just a moment. Okay, let's look at this block of brain. Do you see this yellow stuff here? This yellow stuff is how much dye has moved out into the extracellular space around a set of arterioles that were injected maybe 20 minutes before in an animal that's awake and moving. And here, Eric, this green stuff, they've superimposed exactly the same parameter, the same timing in an animal that's asleep. So this trash collection takes place when you're sleeping. Here it is quantified. Here's how much of the tracer moves into the extracellular space over 30 minutes when you're awake, not very darn much of it, and when you're asleep. And guess what? It's not circadian. If I take Zhu Sheng, who's probably jet-lagged, and I give him some Ambien and watch him for a couple of hours, and he goes to sleep, immediately his extracellular space opens up. This is an animal that's been given a ketamine anesthetic, wide awake, and that's the yellow stuff. I anesthetize the animal, and boom. It's only an hour later, not the normal bedtime for this particular experimental subject. And now, asleep, the space is open, CSF is pouring through. How does that work? What's going on? So first of all, it was necessary to prove that, in fact, the space actually gets bigger. There are some very clever biophysical ways to do that. I won't go into details. Suffice it to say, this looks like a hard data point, this little cartoon. John, you buying this? Here's a narrow vessel, and then look, I drew another cartoon here where the vessel is really big. So, of course, I'm just kidding around. They've done very careful biophysics, looking at an impermeant ion injected into the space and measuring how much accumulates per unit time. When the space is tight, a lot accumulates because the space is tight. There's not a lot of fluid in which it's dissolved. But when the space is huge, now you put the same amount in and the concentration change is marginal. That's how you do this experiment. Okay, very interesting. You've proved that to me, but how in the heck does this thing work? Well, we only have a theory right now. If I asked you to tell me how you awake and how you go to sleep, I hope somebody in the audience would say, well, I remember that the locus ceruleus is a neuroanatomic area with wide diffuse projections, and I remember that those projections are especially active during daytime, and that the released neurotransmitter is noradrenaline. And Mike thought, yeah, that's right. I wonder if noradrenaline does anything. And she's got some provocative data. I wouldn't call this proof, but I would say it's certainly tantalizing. When you take a completely awake animal and give that animal an antagonist of noradrenaline, the space opens up. So here's an animal still awake, but given a brain dosing of noradrenaline, and this animal's space is now wide open. So stay tuned. The implications of this work go in so many directions, it almost makes you dizzy. If you have head trauma, it turns out that you get redistribution of Aquaporin IV. If you get redistribution of Aquaporin IV, there's a sizable chance that this plumbing system won't work as well. And these patients may wind up accumulating, unable to remove beta amyloid and a whole raft of other compounds. It could be old age and some kind of neurodegenerative, perhaps vascular process. It could be playing NFL-style football. There's all kinds of ways that this might come about. Mike and her colleagues have probably published close to 15 papers in the last year and a half on this topic, including a very sexy study with a neuroradiologist developing a prototype of an imaging system that will follow a dye or a radioisotope as it moves through the brain in this fashion. So when we get our patients in 5 years from now, you'll be ordering a NIDA Guard scan, which will include an investigation of this patient's capacity to move CSF through brain and achieve this cleansing function. I'm sorry? She used, I think she used a couple of different well-known noradrenergic antagonists. They usually have about 14 syllables, and I can't remember them. I can't remember them when I've moved from the paragraph where they're described to the paragraph below. So I certainly don't remember them now, but I can get that for you very easily.
Does the volume of CSF production change?
Not that we know of, Phil. Phil's an expert on CSF. He used to give an annual lecture on CSF. It doesn't change too much. You know, there's that law. If you increase the pressure, it goes down a little bit, but not huge. And it's never been investigated with these principles in mind. Okay, white matter rules. So again, I'm trying to be clever. White matter rules as in right on white matter, or white matter rules in we need some rules for the white matter. Tom took it that way. So the question is, you know, am I talking about anything meaningful, or is this just some gobbledygook? Most of you know that your insightful, dedicated, always on the job leaders in Washington, D.C. Have basically invested a lot of money in neuroscience through a program called BRAIN Initiative, Brain Research through Advancing Innovative Neurotechnologies. I think they dumped in the neighborhood of $300 million, or we're planning to. If the Fed, you know, stops printing money, maybe they're going to only be able to put in 50. I don't know about that. And I think it's fair to say about what I'm going to talk about next, that in fact it would be a fantastic thing for us to map every single connection in the human brain. And then to look to see whether Adam's brain is wired the same as Eric's brain and Tom's brain, because they may have some subtle differences in their wiring that would be of interest to you know, those guys in the military. You know, risky stuff. John may have some risky wires that would be good for us to know about. So in any event, this is the birth. Some of that money, $40 to $50 million, is going to be used to create the human connectome. I really hate that they stole that term, because I was going to write a novel about the human connectome. But now I guess I can't. So the point of this is to do this mapping, to use a variety of non-invasive techniques to map every connection in our brains, and to do it not on one brain, which is how we've done an awful lot of translational or human-oriented neuroscience. The whole point here is to do thousands of brains in a highly systematic, absolutely identical fashion, so that we can begin to pick out individual differences, correlate them with unique abilities, you know, the vocalist, the piano player, the banjo player, the neurologist, the ER doc, and so it goes. This is well underway. At this point, phase 1 is essentially done. Phase 2, where they're working on collecting connectomes on 1,200 people, has begun to dump stuff. Oh, by the way, that's the biggest feature of this. You guys can have all of this data you want. So tomorrow, Sumi can take and log in. She has to say, pledge allegiance. I'm not going to use this for any bad stuff. There's a little, some of that stuff. It's much shorter than Apple's I agree thing. And then you have access to incredible raw data. You've got some data that has been rearranged according to certain informatics platforms. You can do some science from you know, your easy chair at home. So they're going to use 4 kinds of imaging techniques: resting state, functional MRI, diffusion imaging, task evoked functional MRI. So they're going to ask, I've been picking on Eric, they're going to ask Phil to play the flute. And then they're going to do some real-time imaging to see how that changes his functional MRI, where the blood is going, et cetera, et cetera. And the hope is that this is going to yield an enormous crop, a wholesale profit of wonderful insights into brain function. In short, it will transform our understanding of the human brain in health and disease. Everybody's getting on board. I got an issue of National Geographic fairly recently that talked about all of this, and the pictures are stupendous. Now, I wouldn't want anyone to leave thinking that those are individual axons. They take data that shows the streaming direction of large white matter pathways, and then, of course, they make them into these strings. I mean, you could count the number of axons here, and I'm pretty sure you can't do that in our corpus callosi. But again, this is kind of a front end of one of those websites. The data is often coded by color depending on direction. So the stuff coming straight up through the brainstem is blue, I think, in this diagram. The stuff going from front to back is green, and the stuff going left to right is red. So you can take a lot of information from one of these things. As I say, the pictures are absolutely amazing. Now, is everybody happy? Have you ever known in the United States that you know and love for everybody to be happy? No, not everybody's happy. So there's been an enormous rhubarb about whether or not this incredible investment is worth the effort. Maybe it's a little premature. Where are the astrocytes? Where are the microglia? Right, so there's been some foot stamping, while opponents claimed that the whole thing might be a gigantic, they said waste, but I'm thinking boondoggle of valuable resources. The same thing is going on in Europe, except Europe is really kind of making us look like pikers. They have invested the same number of dollars, but in euros. So quite a bit more, 50% more. And they're having the same fun conversations.
What are the unexpected breakthroughs that
Which unexpected breakthrough was that?
that were that the EU people found disputes with the EU?
Disputes that continue even as unexpected breakthroughs have begun to surface. You know what? This article didn't tell me what surfaced. And I personally haven't seen anything surface. And I've been watching the surface pretty carefully. So just the other day when I was making rounds, and almost every time I make rounds and talk to residents, I say, why are humans so smart? How in the heck can they come back? Annie gives me information at such an incredible rapid rate I can't keep up. So I'm not saying I'm smart. I'm just saying that some humans are really smart. And that question has gone unanswered. In fact, the presumption has been it's a scaling issue. So your brain is substantially larger than a mouse's. And a mouse is not doing that much. But your dog, oh my gosh, your dog is just terrific. He's got a slightly bigger brain. And then, you know, the pig. Pigs are supposed to be very smart animals. They're about double the size of a dog brain. I have never investigated that personally about the pig. And then our brain. So it's simply a scaling thing. I don't think I believe that. I'm being very analytical and reductionist in this conversation, and I don't buy it. I actually don't think it is just a scaling issue. And dolphins, certain other animals, the elephant, of course people argue an elephant doesn't have as many neurons because his neurons are so damn big. So again, people have kind of hid in these crevices to maintain the idea that it's about scale.
So some people think they're smart.
Elephants.
And dolphins.
Yeah, some people do. I don't know if I do or not, Tommy. We need to talk about that. So a couple of things to start the ball rolling. One of them is that you might have imagined that all the big advances are going to be in the area of what the neurons do, how they're shaped, how they connect, what kind of transmitters they use, their protein repertoire. Well, guess what? Here's a humbling little fact for you to take home tonight and chew on. They use essentially exactly, the mouse uses exactly the same transmitters you do. You weren't smart enough to invent a new one. And in fact, I defy you to come up with a transmitter, just a conventional transmitter that the mouse didn't invent long before we got so smart. There are a little bit of differences in protein expression in the brain. Some of those may turn out to be really interesting, but the humbling truth is that the neurons aren't very different from a mouse to man. But that is not true about the astrocyte. So this is GFAP, so it's simply staining the skeleton of an astrocyte. Here at the same scale is a mouse astrocyte, same scale a human astrocyte. Holy smokes, are you kidding me? That's not the same cell, is it? Yes. 10 times as many processes, 3 times the average diameter. They're monstrous. If you fill them with a dye so that you're not just looking at their skeleton, again, it's crystal clear that the mouse astrocyte looks pretty puny. This was the biggest one we could find, and this is not the biggest human astrocyte we could find. The human astrocyte is enormously larger and more complex. And when you look at the neurons, not so much difference. My colleague, Kinetic Guard, is married to a neurologist, Steve Goldman. And Steve Goldman is a very good neuroscientist who's interested in stem cells. And his work has permitted him to humanize, to be able to capture human astrocytes and then get them to be at a fetal state of preparedness so that he can actually inject them into a mouse. So I'm taking human astrocytes that are expressing what humans express, and I know that for sure because I've marked a protein which is uniquely expressed by human beings, and I've attached it to a fluoroprobe. So every time one of these cells is around, it's expressing a sign that says, here I am, human astrocyte right over here, right here. So he puts a bunch of these in a young mouse, which is a very kind of conducive environment for cells to grow. Does it in, I can't remember, 6 or 10 spots so that they're scattered around, and then he waits. And I'm sure when they first did this experiment, they were stunned when they finally took one of these adult mice and popped it open, and all these white dots, and they are everywhere in the brain, but mainly usually within a millimeter or 2, which in a mouse is a pretty big distance, of where they put these needles in. These cells spread out, and they matured, and the mouse was none the worse for wear. These mice did terrific, and when they looked, they found these huge human astrocytes, which dwarfed the tiny little mouse astrocytes, and they were scattered every place. They made a real point of making sure there were plenty of these human astrocytes applied to the hippocampus. Everybody with me?
The mouse got smarter.
We've got a mouse. We've got a mouse. I will take hecklers out and shoot them. So we've got a mouse, and we've loaded the mouse up with one part of what makes you so special, Gwen, and that is your astrocytes. And now we're going to have a look what this mouse is up to. Well, first I want to make it real clear. Human astrocytes are friendly. They get close to the other astrocytes. They make gap junctions with them. They pass stuff back and forth. They talk regularly. There doesn't seem to be any discrimination. They haven't built their own little town, preventing the mouse astrocytes from coming in. Everything seems copacetic and nice. When they do the staining in a different way, just to give you some sense about the human, you can see they're scattered every place. They're huge. Here's a stain with enhanced green fluorescent protein expressed by these cells. The white shows you it's human, scattered pretty much everywhere. Then they took a look at how smart they were. And they did a whole lot of tests, and I don't have time to go into them. All I can tell you is that they had a battery of about 5 little learning tasks that they could do with mice, and they could reliably get good quantitative results. And the mice, double-blinded, the experimenters didn't know which mice was which. The mice that had the human astrocytes always did better on the learning tasks, and they even did better at LTP, which is something you can do in the dish, where you give a train of high-frequency stimuli, and then the electrical synaptic response remains greatly elevated in comparison to a control animal. So when they do that, here's the baseline reaction. When they do that to these animals that were chimeric with human astrocytes, this was always very significantly, statistically significantly greater. Is this a parlor trick? Is it something that is going to revolutionize neuroscience, and those people in the military-industrial complex will come knocking to figure out what it is about the astrocyte that makes them so smart? Maybe we'll have a military-grade astrocyte that could be used sometime in the future. We don't know yet. Okay, one final little 3-minute story, and then we will be done. I kind of had a feeling that we'd wind up in this condition with about 3 minutes left. And by the way, I took a risk telling you that story about CSF circulation, because now if I ever catch somebody sleeping in the front row of my lecture, they're simply going to tell me, don't scold me, I had stuff I had to get rid of, including the first part of your talk. Out. Okay, so there's 3 more, with this, there's 2 more slides, right? 2 more slides, and you're going to have to think a little bit. So you only have to think, though, for about 3 or 4 more minutes. This is a paper that came out in Cell a while back, but it's still bouncing around, and a whole bunch of other papers have come, and they have largely supported this. And since this is far and away the best example of what I want to tell you about, I've chosen to go back in time a little bit. This is a group. Christina Alberini is at Mount Sinai Hospital in New York City. She is a world-class behaviorist, psychologist. Pierre Magistretti is a world-renowned neuroscientist who works on glial cells and energy metabolism in the brain, and is very famous for the idea that astrocytes will be able to help neurons with energy metabolism by letting out lactate, which can be transported very easily into extracellular space and easily taken up by neighboring neurons. Okay, so that's the group. They're doing a test in animals called inhibitory avoidance. And in inhibitory avoidance, I take somebody from the audience who volunteers, maybe Phil, and I make him stand on a wire grid, which you can plainly see. I painted it bright red, and he can feel the wire under his feet, and I give him a whopping shock. Not enough to knock him down, but he got the message that this is probably not a very good place to stand. He can see 5 feet away there's a place that doesn't have this wire grid, and after I've shocked him one time, I predict, maybe twice, it depends on his mood and stuff, he's going to go over and stand where there is no grid that's painted this bright red color, and if he doesn't get shocked there in a while, he's going to say, okay, I got this thing figured out. So we do this with mice. We shock them, and this latency is how long after we've shocked them that they would actually venture back into that grid area. You with me? So the longer it takes, the smarter they are. They know, oh, I'm not going back into that grid. That's where I got shocked last time. So if I do this, Patricia, she stays in that linoleum area forever. She's just not going back in the grid because, you know, stuff happens on the grid. And that's why this latency is higher. Now, there's a drug I can use which will block the breakdown of glycogen, which is only contained in astrocytes, and which may be part of this system whereby astrocytes can break down the glycogen, use anaerobic metabolism, creates lots of lactate, and dump it into the extracellular space, get it over to the hungry neurons, Maybe they're hungry. We don't know. And it turns out that this drug, DAB, blocks glycogen's breakdown. So when I give the animal this drug at 1 hour, nothing happens. The animal has learned the task completely normally. But if I give the animal that drug there, at 1 hour everything is fine. But if I wait a whole day and I go back and test, the animals that simply got vehicle injected into their brains are fine. They just go about, they remember the task, and by the way, they will remember this task for a week. They don't want to go on the grid. But the animals that had remembered at 1 hour, when they got this drug breaking that blocks glycogen breakdown, they don't remember. They're completely amnestic. They perform no better than chance at staying on the linoleum. So blocking glycogen, which is exclusively in astrocytes, has prevented the acquisition of this memory trace. Can the animals still learn after you've done this to them? Remember, we've got a precise timing. So I've given this drug to block glycogen. It works for maybe an hour. And now this animal can never remember what we did to him. But if I take that animal, the one that I had given DAB to, and test whether he can learn again, no problem. And he can remember. If this were true, I should be able to measure lactate in the extracellular space, and indeed you can. This is an electrode put in the temporal lobe when I train the animal, and with every single successful training event, lactate goes up substantially above where it was before. And here is the final slide. And let's see if I can figure it out. So we've trained our animals. We've given one animal some sugar water into the brain. This animal got this glycogen breakdown, or this glycogen breakdown is blocked by DAB, and he's amnestic when I've done that. This animal also got DAB and can't break down glycogen. But during the time of this test, I gave him some lactate. I injected it into his brain. So, can't break down glycogen, and when I do that just by itself, completely amnestic. If I prevent the glycogen breakdown, but I supplement the brain, I inject, I know it's kind of weird, right, to think about this, but a needle literally goes into the brain, very tiny, squirts in a tiny bit of lactate, it goes up, and now these animals have no problem with memory. Now there's a ton more to this story, but it gets down to the bottom line that astrocytes, glycogen, and lactate appear to be absolutely essential for the long-term consolidation of memory. This work, published in Cell, which is a highly critical journal, very, very demanding. They made them jump through hundreds of hoops. I think they were in review for more than 2 years because it's a little inflammatory to think that the humble astrocyte and glycogen would be so crucial for making memory traces. They pursued this with blockers of lactate transport, et cetera, and it all comes out the same way. Which brings me, okay, the astrocyte, glycogen, blah, blah, blah. Which brings me to my last slide about Albert Einstein. Some of you may have missed this because your reading is incomplete. And just on the off chance that you did miss it, here it is again. This is from the San Francisco Chronicle, a highly reviewed paper. I think maybe they've had 1 or 2 retractions over the last 20 years, but you know, this is stuff you can hang your hat on. And they interviewed Marion Diamond. And Marion Diamond had come into possession of Einstein's brain. Some of you know this story. And she chopped it up, and she found something that she said was very, very exciting. I think I took out one very to get it on the line comfortably. She said, as she explained about the glial cells, that Einstein had a portion of his brain where there were a whole lot more glial cells. Well, I don't think I have to say anything more here, right? My work here is done. And obviously, the demanding standards of the Chronicle being what they are, when they concluded that that might be the reason Einstein was so smart, I've been shocked all these years that that has not made its way into our textbooks. So thank you very much, and if there are any residual questions or thoughts, I'm happy to try to contend with them. Honest with you, Sumi, you know the horse where you put, what are those things called? The blinds that you put on a horse. You do that so the horse doesn't get distracted. Every single one of us wears those. You don't see them, but your vision is limited because you have to stay focused. So it could be that yesterday Gwen passed up $1 million idea because she's got to focus on her work. She can't be thinking about everything. So your question is a terrific one. And I'm perfectly open to the notion that it might in fact be true that chronic problems with mentation, you know, the correlation between the amount of amyloid deposition or plaques or, you know, microvascular changes in mentation are horrible. You can draw a little correlation thing that's statistically significant, but it's a horrible looking graph with points all over the place. So I'm very open to the notion that the thing that ultimately might turn out to have a much better correlation is a slow degradation of the capacity for astrocytes to participate in this complex and very precise metabolic supply chain. And it's got like 10 steps, and it could be interrupted at any one of these steps with, you know, problems resulting. So I like it, and I'd like to collaborate with you. I'd like you to get the grant to fund the work, but I'm there to help. Let's see, is there anything here? Fascinating talk, given association between amyloid angiopathy and dementia. Do you think amyloid deposition could impair the cleaning capacity of the Virchow-Robin space and perhaps cause the dementia associated with amyloid? I do. Next question. I absolutely do. I think that's a perfectly reasonable thing to think about. And of course, it could be very complex, permitting me to talk for another 5 minutes without actually making a point. So I won't do that. But yeah, I like the idea. How would an Aquaporin IV blocker affect the glymphatic system? Okay. So one of the things that I probably didn't do justice to is exactly how this flow in a absolutely you know, fluid dynamic way operates. I mentioned that arterial pulsations are believed to drive the flow from the arterial side over to the venous side. And in fact, it turns out that there's a paper and substantial evidence. I believe that the fluid coming through, making its way over the top of the brain, down into the brain, and then out of the brain, needs ready access to that extracellular space. So the answer to your question, it's kind of weird answering a question and you can't make any eye contact with the person who asked it. But I believe that the Aquaporin IV, which is just absolutely dense on both sides of the astrocytic end foot, which completely and thoroughly, and I mean absolutely, invests all of these arterioles diving into the brain, that without Aquaporin IV, that is materially diminished. It's slowed down, and it may even be a matter of not just slowing down, but being qualitatively different. Maybe if it doesn't move at a certain speed, it's less good at picking things up. I mean, here I'm clearly both figuratively and literally waving my hands. I simply don't know. But
So that system must be very abnormal in the Aquaporin IV knockout.
it must be very abnormal in the Aquaporin IV knockouts. And if that's so, Tom wants to write to the NIH to do a study about these knockout animals to see whether or not their memory and learning might be impaired in the absence of big loads of beta amyloid as they age. I think that's an interesting question. Yes, please.
I thought there was something about the neurons actually shrinking during sleep.
I'm sorry?
I thought there was something about the neurons in the brain actually shrinking.
There's no evidence of that. The only thing that shrinks is during waking. We're active right now. So when we're active, we're not cleaning. We're cleaning when we're sleeping. So when we sleep, and what's the accordion? What mechanism shrinks? I think it's the astrocyte. Now remember, 80% of your brain volume is intracellular. 20% is extracellular. If I change the extracellular space by 5%, I don't have to change the intracellular space by very much. Yet this very substantial change. That's why if you first heard this talk and didn't think about it deeply, you might say, I don't buy any of this. That would mean the brain is a bunch of balloons that are filling up and shrinking. So you have to keep in mind this very unique relationship between the small amount of extracellular space, where a small amount of increase is a huge percentage increase, and what you would have to do intracellular. So I think the astrocyte has the capability to take up that much water for that purpose. So this has been great fun. Thank you so much. And Claire, Claire's not here. Claire's not here. But anyway, Claire, good idea about the 5 new things.